Swayam Prabha. Digital India. Educated India.
It is customary to give a lecture in the RSIC program. Ever since it started, I have been giving lectures in the program. And I always want to give a different kind of a lecture every time I come into the stage here. I don't want to repeat the same lecture over 10 years and then get saturated with the information. So this year I thought I should talk about something nobody talks about nowadays in organic chemistry, namely natural products. Natural products are, organic chemistry can be broadly divided into two groups of compounds: unnatural compounds and natural compounds. Unnatural compounds are compounds which do not occur in nature. And this is a ridiculous statement because organic chemistry deals with nature. So you cannot have an unnatural compound in terms of the structure of the compound and so on. Example of an unnatural compound is given here. This is a molecule known as pyrene. This is tetraethynyl pyrene. There are 4 acetylenic groups attached to the pyrene ring, and it is a highly fluorescent molecule. It doesn't occur in nature. It has to be synthesized. And this is a molecule which looks like a rotor with 6 blades. Can you visualize the 3-dimensional structure of this molecule?
Yes, sir. Yes, sir.
The molecule is viewed from the top, then it looks like this. You have one benzene ring and another benzene ring parallel to it at a distance, and all the 6 carbons are interconnected with respect to each other.
Yes, sir.
And these are molecules which do not occur in nature. This is purely an imagination of the scientist who works on it. The design is important, and synthesis and characterization completes the story there. They are usually applied in medicinal chemistry and materials chemistry, more of material chemistry than medicinal chemistry, because of the pi conjugation that they have in these molecules. Natural products are already existing in nature. All you have to do is isolate it and characterize it. You don't synthesize a natural product unless natural product derivatization has to be done, then synthetic modification of natural products is taken up. After you isolate and characterize the natural product, the only interest that the people have is to investigate the biological activities of the natural products. That could lead to drug discovery. In fact, 70% of the drugs that we have are based on natural products chemistry. You derive inspiration from natural products and apply it to medicinal chemistry, and thereby you come across the drug discovery protocol. So what are natural products? Natural products are products of biochemical reaction. Biochemical reaction take place in living system, and any living system can produce a natural product. In fact, all the living systems that we know of produces natural products. They are primary metabolites. Products are primary metabolites. What are primary metabolites? These are chemical substances which are used as building block for the essential survival and creation of organisms. Whereas natural products by definition is product of secondary metabolites, just like alkaloids, terpenes, steroids and so on. They are non-essential for the survival of the organism. One can have some organism which produces an alkaloid, and one can modify the same organism to shut down the production of the alkaloids. So this is a possibility that exists in nature. We will talk about natural products in this lecture. Natural products can come from any source. It can come from marine source, marine animals, marine sponges, and so on. It can also come from plant-based systems can also produce natural products. These are amphibians. These can produce natural products. And this is a land insect kingdom that also can produce natural products. This is an abbreviation for the various amino acids that are polypeptide chain is what is represented by the abbreviation here. You can see the complexity of the molecules that are produced in these systems. The complexity is quite high and the number of oxygen atom usually is very high. And we don't know why nature produces such molecules and what is the role of these molecules in the living organisms. One cannot very clearly say that the living organism produces these molecules only for such and such purpose.
So natural products are products of both primary and secondary metals.
But people have narrowed down the definition and taking into consideration only the secondary metabolites to be natural products. By convention, this is a...
Sir, I have another question. Sir, how exactly do you read melatonin, like the structure for it? Yeah, but
This is biochemistry basically.
each one is a separate.
Each one is a separate amino acid.
So G will be guanine.
But K is, you don't have an amino acid corresponding to K. The nomenclature is something to do with the Latin names and I'm not quite sure how one arrive at the polypeptide chain like this. Variety of marine sources for compounds used in clinical trials and drugs. These are marine sources, marine sponges, algae, tunicates, and so on. And they produce polyketide, polypeptide, and so on. These are molecules which are natural products. And they are used in the application of anti-cancer drugs and so on. Dolastatin, for example, is a drug that is used for controlling
Sir,
you want to ask something, please go ahead ask.
sir, which is for anti-cancer?
For example, the ones that are numbered, ET 743, AE 941, and so on. This could be Briastatin, for example. This could be an anti-cancer agent.
Sir, anti-cancer means how does it work? Does it does it like kill the cancer cells?
Selectively kill the cancer cells. Not every cell is killed in the
So
process.
is that what is done in chemotherapy?
Yeah. Chemotherapy basically has a cocktail of these molecules, and this is administered to the person. And look for toxicity studies have to be done. Look for the specificity of the drug towards cancer cells, and so on. This is a marine algae. This marine algae produces this particular molecule. You can see the beauty of the structure that you have this molecule. This is called a brevetoxin produced by algae species Carina SSP. It's a neurological shellfish poisoning. So this is a toxicity is there, but at the same time it has biological activity. One can investigate the positive aspects of the biological activities of this kind of molecules. Methyl group. This kind of a molecule is maybe easy enough to isolate from marine algae. In a few milligrams you will get and you can characterize it. But to synthesize the same few milligram in the laboratory is going to be a nightmare, because the number of stereo centers in this molecule, each stereo center is going to have its own enantiomer, mirror image relationship, and so on. So the large number of, once again, a large number of isomers are possible for this kind of molecule, and as a result of that, you have biological activity present in the system. Yeah.
How is the structure of these compounds with a large number of links being found out?
You take the natural source, marine algae, bring it to the laboratory, and do extractions using solvents. And these extractions have to be fractionated properly. One can use HPLC or high performance liquid chromatography, or HPLC, abbreviated as HPLC. And fractional column is done, and each fraction is analyzed for this molecule. When you do a natural products chemistry, you do not know offhand what kind of molecule you are going to get out of nature. So the molecules in the pure form are isolated. The purity is established by chromatography and other techniques. And then the structure determination is carried out using spectroscopic methods. So once you have the spectroscopic data matching the structure, you can be fairly confident that you have arrived at the final product out of the natural product extraction. This is a normal procedure that one does in the natural products chemistry.
So what is characterization?
Characterization means identifying the structure of the molecule. For example, there is a methyl group with a dash line here. It indicates this methyl group is behind the oxygen and the carbon. How do we know this? It's only from the spectroscopic data one can characterize this. The spectrum of this kind of a molecule, NMR spectrum of this kind of a molecule, is not going to be a simple task. It's going to be highly complicated, and one has to sit down and analyze the spectrum properly to get the structure correctly. Coming to terrestrial natural products, this is a flower you can see in the campus. Quite a few places you can see this. These are Vinca rosea. Vinca rosea is the name of this plant, and the extraction of the plant gives these two products, vincristine and vinblastine. These are two natural products. You can again see the complexity of the nature of the natural products in this particular instance. These are, some of you might have seen this in the National Geographic documentary. Colombian Aborigine people actually hunt with the poison dart.
Yes, sir.
Yes.
And the poison dart is poisoned by rubbing the tip of the dart on the frog. So it is highly toxic in terms of poisonous nature of the toxins that are secreted by the frogs on the skin. It's so much so that you can kill 10 persons with one frog's worth of toxin. You can see the structural variation. This is not from the same frog, probably. Different frogs produce different kinds of toxins. They are not the same. And these are not the colored substances that you see in the frog. These are probably colorless substances which are hidden in the under the skin of the frog.
Sir, all these are like common names.
No, these are names given by the scientists based on not IUPAC. one golden frog can produce enough toxins to kill 10 persons, 10 human beings, for example. And how does it become toxic in nature? Because this is a molecule which is capable of shutting down the sodium channels in living systems. You have sodium channels, potassium channels in the biological system. The cell wall has a
Neurological functioning.
not neurological function. Even ordinary cells have the mitochondria, as for example.
How do they produce these toxins?
How do they produce the toxin? This is through biological reaction that goes in their body.
Sir, is it because of their diet or are they born with the ability to do it?
They are born with the ability to do it. That Vincarosia plant, whether you grow it in Chennai or in New York or Moscow, wherever it's grown, it will produce the same molecule. Very rarely changes are seen when the molecules are produced in different regions of the world. So the sodium channel is sort of blocked or opened by the toxins, flooding the cells with the sodium ion. When there is a sodium imbalance, the cell dies. That is how it becomes toxic in nature. And they have come across these two amino acids to be used as a mutation in the DNA of the frog, and thereby the toxicity is reduced or absent in the mutated gene modified frog. Strychnine is an alkaloid. It's a highly complicated structure. There are several stereocenters. All these centers are stereocenters. Stereocenter means it can have an opposite configuration of the system here, and it can have that many number of stereoisomers. If you have n stereocenters, you can have 2 to the power n stereocenters in the molecule. He is right, actually. You can have, what he means by symmetry is there is one half of the molecule has the same set of groups compared to the other half of the molecule. This is a stereochemically active molecule, but it has a plane of symmetry present here. One half of the molecule is mirror image of the other half of the molecule. So this will not have 2 to the power n. This will have a different expression for the.
Sir, is there a formula for it?
Yeah, I forgot now. I don't remember, but there is a formula. This is a strychnose tree, and that produces strychnine. Strychnine is an alkaloid which is heavily toxic in nature, and these two names are written to tell you a story about Stanford University. How many of you know the story of Stanford University?
Sir, their son didn't get admission somewhere and then they started Stanford.
Yeah. The story doesn't end there. After that, Stanford University becomes famous. Leland passes away, and Jane is in charge of the administration of the entire estate and so on. They were quite rich at that time. And Jane also dies suddenly one day, and post-mortem reveals that she was strychnine poisoned. So see how the story connects. And it was not investigated. There was a lot of golmaal went on during the investigation because of money involved. This is a Pacific yew bark tree. The bark of this tree has this particular compound produced, and it is available only in the bark of the tree and not the rest of the tree. So when you remove the bark to isolate this compound, the tree dies actually. So there was a need for synthesis of this compound in the laboratory, and several people got involved in the synthesis of this particular molecule. Successfully they have synthesized the molecule so that you don't have to kill the bark tree. What is this tree? You can see the complexity again of the molecule with the number of oxygen being more than 10 or so here. This is a natural pesticide and insecticide. Indian scientists worked on it and established the structure. There was one person by name Govindachari who spent a lifetime investigating the neem tree alkaloids. And he is the one who finally solved the structure of the molecule using NMR spectroscopy. Azadirachtin based insecticide is used to control various plant damaging insects. Neem seed extract contains azadirachtin, can be used as a natural fertilizer to control the plant diseases. It is explored for the potential use of dental care and pharmaceutical agent. So you can see some of the toothpaste advertisement, toothpaste like neem and Upurika. It's another advertisement. Natural products can come from anywhere. They can come from fungi, they can come from plant sources, they can come from microorganisms, bacterial in other sources. From animals also you can have animal venoms are also having natural products. Vancomycin is a branched tricyclic glycosylated non-ribosomal peptide. It is a mouthful. First derived in 1956 from bacterium, found in the soil of Indonesia and India. Drug is effective against gram-positive organisms, including these several bacterial extracts. Vancomycin is naturally produced by the soil bacterium Amyclotopsis orientalis. These all sound like asterisks and obliques names, right? When I was reading this is what came into my mind. This is the structure of vancomycin. Just to kill a bacteria, you need to have such a large molecule to. Sorry? You can count the carbon and oxygen and see whether you can come up with the right structure of the. As an exercise, you can do that. What is this plant? Opium, poppy. Morphine is a major constituent of opium. Derivatization of morphine gives what? If you esterify this to hydroxy functional group and make a ester out of it, you get heroin. Morphine has this 2-hydroxy functional group that is esterified into the acetate ester. That acetate ester is the one which is known as heroin. These are narcotic substances with beneficial anti-inflammatory and anti-aching properties. Streptomyces zelenesis is the culture from which this molecule was isolated. This looks like a necklace actually. It is initially isolated in trace amounts from Streptomyces zelenesis in Upjohn. Upjohn is the name of the company in 1978. That is why they are given a number rather than a name. Companies don't give names to compounds which they discover. They give numbers so that the number is known only to the people who are working in the company. So, mechanism of action is specifically binds to modifying DNA leading to cell death. Application is potential anti-cancer drug due to the high potency and unique mode of action. Unique mode of action is that it intercalates with the DNA and cleaves of the DNA. Zaragosic acid. Zaragosic acid is a family of potent fungal metabolite that acts as a competitive inhibitor of squalene. Squalene synthase is a enzyme which synthesizes squalene. Squalene is useful for synthesis of cholesterol. So cholesterol synthesis will be affected if you take zaragosic acid, for example. It is crucial for the synthesis of cholesterol as well as ergosterol, which are steroids which are used in our body, for example. Zaragoza is the name of a town, city in Spain. Zaragosic acid comes from there. Death of Socrates. This story you must know. He was not buried alive. He was poisoned, slowly poisoned. He was given the extract of hemlock. And why was he killed? What was the reason? Pardon me? Why did he poison himself? Death sentence was given. Why is death sentence was given? He promoted the heliocentric nature of universe, which was against the Catholic Church's geocentric. So he was given the death sentence for disobeying the Catholic Church, for promoting the wrong science according to the Catholic Church. This is an active substance in hemlock tree. Conine is the active substance, which is a piperidine ring with a propyl group attached to it. Penicillin story again. Alexander Fleming accidentally discovered the some spots were inhibited by the, and this is a molecule that is responsible for the inhibition of antibiotic nature of the turmeric, a medicinal plant. It has antiseptic activity, anti-cancer activity. The derivatives of these molecules, which are active ingredients of manjal, the derivatives of this molecule is being investigated for anti-cancer activity. This molecule itself is toxic towards human cells when it is taken internally. This is mostly for topical application only we have. On skin apply. Don't take it in. I mean, this is not toxic in the level of toxicity that we are talking about. Hemlock is of the order of magnitude higher toxic. This can be taken in gram quantities and still you can live. This is used in food preparations, for example. Turmeric is used in food for two reasons. One is for taste, other one is for yeah. Now come to the terpenoid molecules. Terpenoids or isoprenoids are small molecules which occur widespread in nature. They occur in natural products. They show a range of pharmacological activities such as antiviral, antibacterial. This is like a Sarvaroga Nivarani kind of a thing. What is important is 60% of all natural products are terpenes, terpenoids. So they are occurring in nature very frequently compared to the other class of alkaloids and steroids and so on. The basic building block is isoprene. Isoprene is methyl butadiene. This is isoprene. 2 units of this is called monoterpene. 3 units of this is called sesquiterpene. 4 units of this is called diterpene, and so on. This is a ginger plant. Ginger produces this class of compounds. These are essentially same as manjal. If you look at this molecule, 1/2 of the molecule is what the ginger produces. But the properties of these molecules are entirely different. Ginger is pungent and it has its own karam and so on, whereas manjal doesn't have that. Constituents of many essential oils are terpenoids used in perfumery and aromatherapy. Insect pheromones are basically terpenoids. What are insect pheromones? These are small molecules used as neurotransmitter in the insects for communication purposes. A male moth can find a female moth through the smell of this kind of pheromones allow them to communicate. Chemical fragrance. When I typed musk in the Google, Elon Musk came like a ton of bricks. This used to happen to me when I was the beginning of my career here. My name is Shankar Raman. Somebody got murdered with the same name. So whenever I search the internet, that Shankar Raman will come. Shankar Raman's death and the case that goes on, so on. This is true actually. My name is Shankar Raman and the guy who died also is Shankar Raman. These animals have their own glands to secrete substances which are highly desirable smell in the sense they are used in the perfumery industry. This is a natural muskone. This is produced by the musk deer through its glands, and it is a stereoisomer of the methyl substituted macrocyclic ring system. While investigating the explosive nature of nitro compounds, accidentally the scientist came across this molecule which smelled like musk. Although there is no structural correlation between these two structures, this is a musk-like compound. So now the perfumery industry is restricted using animal products actually. They cannot use musk because of the cruelty to the animal and so on. So they depend upon the synthetic musk. These are synthetic musks, nitroxylene and polycyclics. There was a time in India, the scientists were very active in natural products chemistry. And we were pioneers in many of the natural product synthesis and isolation, characterization, and so on. Due to wrong policies of the funding agencies, let us say, that particular area has slowly vanished, and nowadays nobody is working on natural products. These are some of the doyens of natural products chemistry in India. Asima Chatterjee is the first woman to receive a PhD degree, I think, in India. Dr. T.R. Sheshadri is a Delhi University professor. He was very fond of growing plants which gave flowers. Flowering plants is what he used to grow in his garden. And he was fascinated by the color produced by these plants and started working on the extraction of the petals of the flowers. And he isolated many natural products which are called flavonoids. And he was quite active till he died in 2001 or so. Professor T.R. Govindachari is a famous person for neem extract and the products associated with neem. What happened here? K is K Venkatraman actually. Venkatraman has disappeared somehow.
Sir, there.
Ramnath is hiding Venkatraman. K. Venkatraman has a name reaction named after him. Baker Venkatraman rearrangement. He was quite famous at the time when he was doing this chemistry, and he was responsible for several flavonoid chemistry as well. Ramnath was a medicinal chemist. He used to take the natural products and test them for biological activity. It's not enough if you do isolation and characterization. One need to identify the biological activity also, whether it is anti-cancer or anti-anemic or whatever the product, whatever the effect it has on the human body, it has to be investigated. B.B. Day and many others worked on natural product chemistry in India. I would like to salute them for their contribution. We should all be proud of their contribution, at least to know some of the contributions they have made. Thank you very much for your attention.
It is with great honor that I would like to present my thanks for the extremely informative lecture. It drew a lot of curiosity from me and many others, I'm sure, about the chemistry of natural products. Accompanied by stories like that of Jane and Leland Stanford and the death of Socrates, made for a truly amazing and knowledge-filled lecture. Thank you, sir.
Swayam Prabha. Digital India. Educated India.